What's good? 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 What's good, everybody? Welcome back to Belvedere ASMR, and welcome back to my lab. This is my tissue culture lab, where, if you don't know, I'm currently doing a PhD. So this is the second video that I filmed here, and I thought this time I could do a similar thing, a bit of a sound assortment with some items around my lab that I think could make some good sounds. But I thought this time I could also include some rambles about what I'm actually doing my PhD on, and like the kind of things that I do day to day. Hopefully that will be interesting, and also, of course, hopefully it will be relaxing. It's a bit weird going back to filming with the small mic, but hopefully it still sounds good. So this time I've put a couple more boxes so that I can, like, you can actually see the mic. But anyway, without further ado, let's get started. Of course, got to start with the gloves. So today is a Sunday. It's currently about 6 p.m. and I had to come in to look after my cells. So I thought I'll bring the mic along, and there'll be no one here, so I can get some footage. I actually wanted, so this is the tissue culture lab, and there's like the main lab out there, which is much bigger, and I really wanted to film a video there. Because it's like a different location to where I filmed the last video. But unfortunately, I was the only person in this whole building for the whole day that I was here. And then I finished all my work, so I was like, okay, I'll film a video. Took the microphone, set everything up, and then these two people turned up. So I had to pack up and come into this room. But it's okay. We'll still get some great sounds for you guys. So I'll just find the first item, and then I can begin telling you about what my PhD is on. So what should we start with? You know what? Let me get some of my actual cells. So here we have a flask of mouse cells. They are mouse fibroblast cells. And basically, so let's start at the beginning. My PhD is on stem cell biology. Now, if you don't know what stem cells are the cells in our body that make the other types of specialized cells. So things like skin cells, heart cells, liver cells, all these different types of cells. They have to be made, so these are specialized cells with specific functions, and they have to be made by these other cells called stem cells, which are like generic cells that can be directed down different routes of differentiation to become the specialized cells. So for example, when you're first like an embryo, when you're growing, all the cells in the embryo are stem cells, because you're not going to have like skin cells in an embryo, because you're an embryo, don't have any skin. You just have the stem cells which grow and grow and grow, and later on will turn into skin cells. Anyway, so stem cells are crucial for things like response to injury and regeneration. And also, they are important in aging, because as people age, the function of your stem cells in terms of their ability to keep reproducing and differentiating into well-functioning specialized cells, as people get older, it declines, and that causes age-related diseases. So my PhD is focusing on stem cells in the human lung, and therefore focusing the bigger picture is trying to find treatments for chronic lung diseases, such as chronic obstructive pulmonary disorder, pulmonary fibrosis, and other such diseases that are a particularly big problem in older people. Okay, I don't want to type on this too much because the cells will start coming off. This is Dalbeco's phosphate buffered saline. So basically, we know that stem cells in our lungs are crucial for everyday regeneration. But we also know that as we get older, their function declines, leading to these chronic lung diseases. What we don't know is what actually changes in these lung stem cells as people age. It's never really been researched what actually happens in these stem cells. And so that is what I'm doing, or going to try to do. I'm going to be looking at aging in these lung stem cells and potentially finding genetic targets that can be used to create treatments. So what that means is I'm going to be comparing the regulation of gene expression. So what that means is, in our DNA we have thousands and thousands of genes, about 20,000. But of course if every cell has exactly the same DNA, in order for us to have different types of cell, in these different types of cells you need different combinations of these genes to be either like turned on or turned off. And the control of which genes are turned on and turned off is called gene regulation. So I'm going to be looking at how this gene regulation changes. So so what genes are turned on and turned off, how that changes where, like, as humans grow from children to adults to elderly people. And that is with the hope of finding things. So for example, maybe there's certain genes that are very highly expressed, so they're always turned on in like child lung stem cells, and as people get older, the expression of this gene is reduced, leading to a reduction in the stem cell function. So then maybe you can then create drugs that target this gene in the elderly people to increase its expression and improve the function of their stem cells and reverse the effects of these like chronic lung diseases. So that's the aim. And I'm going to be doing this. The reason why I work a lot in this room, the tissue culture room, where you grow cells in those dishes. So yeah, you grow cells in those dishes that I got out, is I'm using human cells and I'm growing them, and I'm using that as like a model. So I'll grow lung stem cells from children, taken from like hospital, and lung stem cells from adults and from elderly people. And then you can do experiments on them to compare the gene regulation and identify any differences. So that's what I'm going to be doing for the first year. And then once I've identified the differences, I'll then do what's called like functional experiments to see, okay, so there are these differences. What happens if I do these changes in the elderly cells? So if I increase the expression of this gene, will that improve the ability of the elderly cells to like differentiate in cell culture, or the ability to proliferate or like grow in cell culture, things like that. And so the way you grow human lung stem cells is you grow them on the mouse cells. So those cells that I had, they are mouse fibroblast cells. Now fibroblast cells are a type of specialized cell that are like structural. They play a structural role, so they kind of support the function of other cells. And so basically you have to plate these mouse cells, and then you plate the human cells on top, and the mouse cells help the human cells to grow. And so you grow them in, you have like all kinds of different sized culture vessels, depending on how many cells you need for your different experiments. So this is a 6-well plate, because you have 1, 2, 3, 4, 5, 6 wells. And so in this, you would plate about 200,000 cells and expect up to 1 million when it's time to like take them out of a well and put them in something else. These scissors don't make a very nice sound. I feel like tapping with gloves makes everything a bit harder because it like dampens the sound. So basically day to day I have to do a lot of cell culture, and you do them in these, oh you can't see those ones, in these hoods, which have like an airflow, and it filters the air so that you only have sterile air in there. Everything has to be very sterile, because if you get infections it messes everything up, everything dies. So yeah, I digest them, because they're like little chunks of human lungs. I have to digest them and then grow them and then freeze them to be used. And then alongside that I do these experiments. So at the moment I'm doing things like, one of them is called ChIP-seq. 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 It stands for chromatin immunoprecipitation sequencing. Now basically, in your cells, your DNA isn't just DNA. It's wrapped around proteins called histones to form something called chromatin. So the combination of DNA and the histone proteins all wrapped together is called chromatin. And one of the ways that your cells controls gene expression, so which genes are on and which genes are off, is through the regulation of the structure of chromatin. Because some areas of DNA, the DNA can be wrapped really tightly around the histones, which means they're not accessible to the things that cause them to be turned on. Other areas, the association between DNA and protein is much more loose, meaning the DNA is accessible to all these different enzymes which can come in and express the genes that are there. So this ChIP-seq basically lets you take your cells and through some very clever experimental procedures and a lot of computer analysis, you can identify specific protein associations across the entire genome, if that makes sense. So you can see where a specific protein will bind along DNA. You can see where specific histones are bound to DNA. And so I'm going to be doing this with a protein called p63, which is like a master regulator of gene expression. So I'm going to be looking at where p63 binds on the human genome in these lung stem cells. And that will help to give me a clue as to what genes are potentially being expressed. Something else that will help me do this is something called ATAC-seq. ATAC-seq. I'm not sure, I can't remember what this stands for. But it lets you identify how accessible different regions of DNA are. So when you are finished analysing it, you get your long sequence of DNA and you get peaks. And the higher the peak, the more open the DNA is. So the less strongly bound it is to the histones. And so these open regions are regions where the genes that are within them are likely to be expressed. Then I'll also be doing something called RNA sequencing. Now when a gene is expressed, the DNA is copied to form something else called RNA, which is a very similar molecule, but instead of having one long thing of RNA like you do DNA, where DNA contains everything, you'll just have one gene's worth of RNA, and these can be turned into proteins. So RNA sequencing, you can take your cells from culture, lyse them, so basically dissolve the cells so that everything inside is released, and then you can isolate all these RNAs and sequence them, and that will tell you exactly which genes are expressed. press the regulator. So that is what I'm working on. I don't know if it made any sense. I tried to explain it. I tried to explain everything as well as I could. But if it doesn't, then let me know and I can try and explain it better in a future video. But yeah, I'm currently 5 months into the PhD, so I haven't done loads yet. But it's definitely exciting, and it's been so I finished undergraduate in the summer, last summer. So it's been a nice switch up. Like, it feels more like I have a job, even though I'm technically still at uni. Whereas at uni, it definitely felt like I was at uni, even though last year I was doing like a master's project. Let's try tapping on this. I was trying to think what kinds of videos I can do here. And I don't know if it's a stupid idea, but I thought it could be kind of funny. But I could do a role play where, like, you guys are myself, and I can pretend like I'm, like, passaging you, so changing you from one flask into another flask, or doing some kind of experiment with you. I don't know. I don't exactly know how that would work. All I know is the camera would be low, and I could put a waste disposal thing just behind you, so that when I'm dispensing media and whatever onto you, it's going just behind you, but it might look like it's going on you. And then of course I could do loads of like sounds with the equipment and stuff. I don't know. It could be a fun idea. I don't know if it would be relaxing though. Sometimes I forget that, like, these videos are made to relax. But they also are like fun as well. But yeah, I definitely don't only want to do like sound assortments. I think there is definitely a lot more potential here. I just need to come up with the good ideas. What else can I make sounds in? This is just a tube holder that you put in a centrifuge. So you put tubes this kind of size in like this, and then it goes in the centrifuge and spins. And that will basically make all your cells go to the bottom and form a pellet, which is useful if you want to change them from like one kind of media to another. these are what you freeze the cells in. Don't know if that makes the first sounds. We of course have the chem gene. Everything around here is going to be super clean after this. I'm not going to spray it too much because I'm pretty sure it's carcinogenic. So I'm not trying to fill the air with it. It's like an incredibly strong cleaning agent. It's like Dettol, but on steroids. This box should, if it's not empty, contain culture slides. Except I think it's empty. Oh no. It's not. It's not, but I probably shouldn't open the other side. Apparently my phone's about to die. Okay. Maybe that's a sign for me to end the video. I hope that you have enjoyed it. I hope that you found the ramble about my PhD interesting. I think a couple of people have suggested doing a video like explaining what I'm actually doing. Yeah, I'd be very happy to continue to talk about the PhD if you would be interested in it. So just let me know, leave a comment below and give me some other ideas of lab related things that you might want to see. But other than that, thank you so much for joining me. Leave a like if you liked the video. Subscribe if you're new. And of course I'll see you tomorrow, where we're actually nearing the end of ASMarch. But still plenty of videos to come. Thank you again for all your support. We are nearing 750 subscribers, so almost 3/4 of the way to 1,000, which is incredible. I need to think of something to do for when I hit 1,000. A little 1,000 subscriber special. So give me some ideas if you have anything that you would want to see for that. But anyway, thank you for joining me. I hope you found it relaxing. And I will catch you in the next one.